Welcome to the July CGC webinar. I'm really excited today to introduce Dr. Gamze Gürsoy. Dr. Gürsoy is an assistant professor in the Department of Biomedical Informatics and Computer Science at Columbia University, and she's also one of the core members at the New York Genome Center. Dr. Gürsoy's lab's overarching research interests or research goal is to harmonize the diverse fields of biology, bioinformatics, molecular biology, and engineering and cryptography with the goal of achieving two fundamental issues: increasing biomedical data access to a wider group of scientists while preserving privacy of research participants, and uncovering the molecular underpinnings of gene dysregulation via knowledge gained from functional genomics data. And Dr. Gürsoy is currently leading a group of computational experimental scientists, and today is going to present some work that she performed with Dr. Graham Irwin on the Cancer Genomics Cloud.
Thank you so much for the kind introduction and also for the invitation. Today I am going to talk about the work with Graham that we've done when we were both postdocs. He was at Mike Snyder's lab at Stanford, and I was at Mark Gersten's lab at Yale. This was a really fun project that us and few undergrads coming together and putting it together. And the most important thing is that it started with a grant from Seven Bridges to use the cancer genomics cloud to basically run our pipelines on many cancer genomes to discover recurrent repeat expansions in cancer. So for that, we are also very grateful for the funding and for also the easy to use platform. So let's start with what a short tandem repeat is. Probably everyone knows what it is. It's basically a repeating motifs in the DNA, these stretches of motifs. Those motifs are usually 2 to 6 base pair. Here we are seeing a repeat that is composed of the GAAA motif. So in some individuals, you might see just 4 repeats of this motif, and in some individuals, you might see 6 repeats. So these are basically repeating elements, and these are called short tandem repeats. So sometimes these tandem repeats get expanded, and you see many copies of it in a single repeat. And it causes over 40 human diseases, most of them being neurodegenerative, such as Friedreich ataxia, Huntington, Fragile X syndrome. So we see these expanded repeats in different regions of the genes and non-coding elements. The repeating elements that causes diseases are usually really large. So this N can go to very large sizes, and then this causes many different diseases. And the mechanism of action for different diseases can be different, but it usually results from these really large expansions. And so I wanted to show one example of how it works. Basically, this is the FRDA that causes the silencing of this gene, causes Friedreich ataxia. And the protein product of this gene is this protein called frataxin. So in normal genomes, you see perhaps 20 copies of this GAA repeats. And in Friedreich ataxia, this copy number can go up to 500, and it's in the intron of this gene. And because of that, the frataxin cannot be expressed. And this was this was something that Graham studied before studying the cancer. And the idea of looking at repeats in cancer came from this project, basically. So it is known that this repeat is I'm not, so I might sound wrong here because I'm not a biologist, but this is because the RNA polymerase cannot pass through basically when you have many copies, and because of that you don't see the expression of the gene. And in Graham's previous work, that they designed synthetic transcription factors that can bind and then allow RNA polymerase to pass through and basically express frataxin. So because of that, we actually wanted to look at the cancer genomes because it was never done before. People did not look at the large repeat expansions in cancer. And the idea is that we actually know that there are these microsatellites, which are also repeating elements, and they are hallmark of several cancers. In certain cancers, you see a lot of microsatellites, and they are being unstable, meaning that they get mutated. So the microsatellite instability happens when you have contraction or expansion of a short repeat, but these are usually very small ones. The repeat expansions is a lot larger than microsatellites. So because of that, we first asked the question whether these microsatellite in stable high cancers also have high rate of recurrent repeat expansion. So that was the first question that we wanted to look at. So basically, we are looking for somatic expansions. And luckily, we have a really nice data source, Peacock data, where we have around 2,600 cancer genomes with matching normals. So we can actually compare if any of these repeats are expanded or contracted in cancer genomes by comparing normal and tumor tissue. But there's a little bit of a problem to do that because the Peacock data is coming from short reads, and short reads have a certain fragment size. And if the repeat is larger than this fragment size, then the reads that comes from the repeat cannot be mapped to those regions. So we were looking for a tool that actually can use the short read whole genome sequencing data because we have all this data and we wanted to look at the repeats. And another reason why we are interested in the repeat expansions that are larger than fragment sizes is because most pathogenic repeat expansions are very large. And if you look at the, ataxin C9orf, which is in ALS, they are larger than 10,000 base pairs. So if the hypothesis is there are some pathogenic repeat expansions in cancer, we expect them to be larger than the fragments that we get in short read sequencing data, and that's why we were looking for a tool that can do that. But again, luckily, there was a tool out there. This is our collaborator Igor and Mike Berle. At the time, they were working for Illumina, and now I think they're at PacBio. So they developed this tool called Expansion Hunter de novo that finds the de novo repeat expansions in a case control genomes. And it does it by basically classifying the reads into different groups, such as the anchored in repeat reads and unpaired in repeat reads. And if we can use anchored in repeat reads basically to figure out the expansions. And paired in repeat reads cannot be used because we don't know where they map, but because they will map to the same location, we can actually use that information to figure out if there is an expansion or not. So it works. So what happens is that if the repeat size is smaller than the mean fragment length, because we are looking at anchored in repeat reads, we are able to accurately capture the repeat length. If it is larger than the mean fragment length, now all these in repeat reads are not mappable. We don't know the size of them, but we know that there is a repeat expansion. So we can accurately say there's a repeat expansion, but we cannot accurately tell the size after 450 base pair. But this is great because we will be able to say if there is a repeat expansion or not in cancer genome. We just won't be able to say exact length using this. So then they also do something very clever because these, the in repeat reads are piling up under the same region. Then that means that you're getting this high sequencing depth there, and using that high sequencing depth, you predict whether there's an expansion or not. But you need to also do some kind of normalization with respect to rest of the depth in the genome. And by doing that, you can avoid basically false positives. So that's Expansion Hunter de novo in a nutshell. I probably didn't do a good job of explaining it, but you can go to this paper. It's really nice, very nice tool, very easy to use. So we decided that we are going to create a pipeline using Expansion Hunter de novo because this was also done shortly before this study started, that autism spectrum disorder genomes, that they use expansion hunter de novo to capture repeat expansion. So we knew that it was working, and you know, also in their paper, they have a lot of validation of these repeat expansions. So we had confidence that this will be able to capture what we are looking for. So we did a small change. So we had 2,500 patients, 29 cancer types. We use expansion hunter de novo to get a initial set of you know, expanded repeats. So as I mentioned, that we have global normalization in expansion hunter de novo, but here, if you look at the cancer genomes, you see a lot of copy number variations, you know, amplifications. So global normalization may not work. So you might be able to seeing this high depth repeat expansion because this region maybe have high copy number variation, and that's why you're seeing this. It may not be just because of the repeat expansion. So we also employed a filter which is based on local read depth normalization, meaning that you look at the tumor's depth, sequencing depth around the peak that you're seeing the repeat expansion, and you normalize it. For example, here I'm showing a false positive. No, this is not a false positive. This is a true positive. I'm showing that how we bring the local depth to the normals depth to figure out, because tumor had a copy number variation around this region, so we can normalize it with the normal sequencing depth, and then now we can confidently say that there is actually a repeat expansion. So that was our addition to the expansion hunter de novo, and this is the complete pipeline. And we packaged it and containerized it, and then put it in the 7 Bridges Cancer Genome portal, so that we can just run it in all the data that we have there. So initially we captured 285,000 loci that had de novo tandem repeats. And then we did the global normalization that expansion hunter de novo suggests, and then we reduced that number to 578, so get rid of all the false positives. And then after local normalization, the total number of recurrent repeat expansions that we captured is 160, and we captured it in 7 different cancers. And here how the distribution is. Some of the repeat expansions were seen across different cancers. Some of them were just belong to a single cancer. And some cancer types had really high number of recurrent repeat expansions, while some cancers didn't have many. And you may ask then, how does it look in a healthy population? So our collaborator also ran this tool on 1,000 genomes projects, genomes from 6,500 genomes. And as you can see that you don't actually see a lot of recurring repeat expansions in healthy population. So I mean, this number looks 160 looks small, but it's actually pretty high when you look at the regular population. So this was kind of nice that you know, we did all these false positive cleaning, and we had this small number, but we knew that it is cancer specific basically. Here I'm showing the frequency of the repeat expansions per different cancer. So as I said, some cancers have a lot more recurring repeat expansions per sample, meaning that per patient. For example, more than 25% of prostate adenocarcinoma has the patients has more than 10 repeat expansions, and in some cancer types you only see 1 repeat expansion per patient. So this is also interesting. And then we can go back and ask the question, well, there was these cancer types that are microsatellite unstable. We wonder if this frequency actually correlate with that. But before that, I will show you that we also looked at some GWAS loci. For example, there's a prostate cancer risk locus, and then we looked at if any of these repeat expansions are close to it, and then we always compare it to simple repeats, basically, as a, you know, this is what you would expect from repeats distance to the nearest cancer risk locus, and then this is what you get basically from the recurring repeat expansion. So we saw that there is they are closer to the cancer risk locus. So, but we don't know, of course. We just know that there is some localization there. We don't know if this is the disease causing repeat, or it's just happened to be close to the cancer risk locus. We also looked at the motif length. As I mentioned in the beginning of my talk, most of the motifs are 2 to 6 base pair, and that's what you see here in cancer as well. And there's a spike with 4 letter motifs, and then there's this bimodal distribution where you also see a lot of motifs that are really large. And then if you look at the fold enrichment, that we see a lot of GAA, GAAA, and ATCCs in these motifs. GAAAs are interesting because in neurodegeneration we see a lot of GAA repeats, so they are kind of similar to that. So this was the motif analysis. And then now I can go back to and ask the question if there's an association with MSI high cancers. And interestingly, you don't see a lot of repeat expansions in cancers that are microsatellite unstable. So those cancers have shorter repeats, and then these other cancers that are not microsatellite unstable, they have larger repeat expansions. So that was kind of interesting, and then we were just going crazy and doing all sorts of statistical tests to make sure that we are correct. Here I'm showing that the microsatellite mutation rate, there is no difference between genomes that doesn't have any repeat versus have repeat. And if you look at it, microsatellite unstable high cancers, you don't actually see any recurrent repeat expansions. And in microsatellite stable cancers, you see some. So there is basically no association whatsoever. So it's interesting, and I wonder if there is a different mechanism that that this is happening. So we, of course, look at where these recurrent repeat expansions are located. We see a lot of introns, which is interesting because, again, I showed you the Friedreich ataxia example. That was also an intron. So that's kind of interesting. There are not many exonic ones. And we also see some on the promoters, but these are well-defined promoters. We also looked at candidate regulatory elements, which I will show in a minute. But yeah, the looking at introns were interesting, that these repeats happen to be on introns, just like in other some of the neurodegenerative diseases. So the next, we looked at if these repeats are on candidate regulatory elements or close to them. Let me explain what candidates is regulatory elements. This is from the ENCODES catalog, where both of our labs, previous labs, postdoc labs, were basically part of ENCODE consortium. And then in this consortium, using all sorts of functional genomics data, the consortium defined these candidate cis regulatory elements, meaning basically there are elements that are regulatory elements that are proximal to the genes, or distal to the genes, or they are on the transcription start sites, basically more like a promoter-like elements. And these elements are derived by looking at different histone modification signals that tells you which regions are active, which regions are repressed, and then also CTCF binding, and also chromatin accessibility through DNase. So basically, if you have all of these, then you're more likely to be a regulatory element. And then if you have some of them, and depending on where you're located with respect to the genes, you can be either a proximal element or a distal element. So the reason why we asked this question is because we were wondering if there are any recurrent repeat expansions basically perturbing the enhancers or promoters and then causing gene expression disruptions. So if you look at their distance to the candidate cis regulatory elements, they are significantly closer to them compared to the simple repeats. Some of them are sitting on top of the regulatory elements, some of them are just near them. So we don't know if there's anything functional. We didn't do any functional study, but they seem to be close to the candidate cis regulatory elements, basically. And then there was this paper in cell, and then before that in neuron, where they were talking about how TP53 is critical for mediating the pathogenic effects of repeat expansions in both ALS and Huntington disease. Both of these diseases are due to repeat expansions, and then if you have TP53 in wild type, not mutated, it seems to be critical. So then we ask, okay, TP53 is one of the cosmic genes, one of the cancer genes that we know. So now we ask the question, is there any kind of association between the occurrence of these recurrent repeat expansions and the cosmic genes? So we looked at around, I think, 500 cosmic genes. Here I'm showing the top 50 mutated genes in cancers, and looked at basically if there is any kind of association. And how we do it is a very, very simple Fisher exact test. We have patients with mutation in a cosmic gene. This is per gene. So we do this test per gene. And then patients without a mutation. And then their overlap with the patients with at least one recurrent repeat expansion, and then patients with no recurrent repeat expansion. So it looks like this. So you have basically a Venn diagram. You're looking at which set is basically happening more, which set has higher propensity to happen with the other set. So basically here I'm showing that here all the patients with a mutation on TP53, these are all the patients that has recurring repeat expansions, and then this is their overlap, and then this is the overlap between the patients with no recurring repeat expansion and with mutations in TP53. So we do this for 500 genes and then correct for multiple hypothesis testing, and then ask what is the p-value of these associations. So what we found is that if there is a high probability that these recurrent repeat expansions are in patients with wild type cosmic genes of these, and these are the only significant associations. So basically what this is saying that if you have a patient and the patient has the wild type TP53, that patient has a higher probability to have a recurrent repeat expansion in their genome. So there is a negative correlation. And it's really interesting that these are really, some of these are famous, highly mutated genes in cancer. So if you have those mutations, that you have less probability to have the recurrent repeat expansions. So again, going back to which cancers that we have, these repeat expansions, just to show you basically that we did some also validation. So for example, we found 2 recurrent repeat expansion in kidney RCC, and then we asked the question whether we can capture it in the wet lab, right? So the data is telling us, but also keep in mind that these are patients from PCOC, and here, before actually talking about that, I want to talk about why we care about this, is because there is this gene UGT2B7. We saw that in the intron of this gene, 34% of the renal cell carcinoma, we actually see this GAAA repeat expansion. And it is overlapping with some, functional genomics signal, but most importantly, it's also close to this distal enhancer-like sequence from CCREs from ENCODE. So it's pretty close. But this distal element is likely not regulating this gene. But regardless, we wanted to figure out if this repeat expansion is real or not. We've been seeing it. So for that, we basically looked at a bunch of cell lines, and so here I'm showing the bands. So this is when you don't have any repeat, you expect it to be here. And some of these cancer cell lines, you see that there are different copies of this GAA repeat expansion. So, and it's interesting because we saw this in patients, and then here we are showing cell lines, and you still see on this gene. this is a very significant repeat expansion that was showing up after all sorts of, false positive cleaning. So that's why we were curious. We also show that, for example, with a cell line that we didn't see this repeat expansion versus with a cell line that we saw this repeat expansion, we did PacBio long read sequencing, and it was very clear that basically these are the cells that no repeat expansion, and this is the cell with repeat expansion. That it's like clearly there is a repeat there. So both experimental validations that they were showing us. And then Graham went to Stanford Med and found some actual samples from kidney cancer patients from the hospital, and those are completely separate test set than the patients that the patient genomes that we use in Peacock. And we were able to see 1, 2, 3, 5 patients out of 12 patients that they had repeat expansion exactly on this gene. We still don't know what it does to the gene. We showed that there was a difference in transcript, the different isoform expression, a little bit difference. But we really don't know, is it that this gene is being perturbed, or maybe this intron is a regulatory element for some other gene, and some other gene is being perturbed. We don't know what is happening functionally, but it is really interesting that this repeat expansion keeps being really persistent in whichever way we look at the data, we see it, and whichever test set we have, we see this repeat expansion. So this is the bit that basically, oh, I'm so sorry. This is the bit that Graham did, and I can't really very well explain, but it's very interesting. So what he was asking is whether these GAAA repeat expansions confer a selective advantage to the cell. So he designed this synthetic TFs, here the SynTF3, that is targeting this GAAA repeat in these cell lines with and without the repeat expansion. And so basically what it's showing that if you give this SynTF3 to the cell with the repeat expansion, then you see that your relative cell density is getting lower compared to the control. So it's really interesting, and he tested in 6 cell lines, and that he can actually really target these repeat expansions, and then shows that the cells actually change the behavior when you do that. So this is the last bit of the paper that I'm not very good at explaining because I'm not a chemical biologist, but you can go to the paper and read it, or contact Graham if you need more. And his previous paper in Science about the Friedreich ataxia was also using a similar approach. Yeah, with this, so it's a little bit of, it's an unfinished work. We haven't finished it yet, and I just wanted to show what the next steps are. So my team largely develops tools, computational tools. So we wanted to develop something that we can also not only capture the expansions, but also tell what the lengths are for people to use in different diseases. And as I mentioned, this work wouldn't have been possible without the funds and the platform that Seven Bridges Cancer Genomics Cloud provided us. So we are very grateful. We also got credit from AWS. That was also great. We're very thankful. Both Graham and I, we both are funded in our postdoc funded through NHGRI K99R00 award. And we are very grateful to Peacock for putting all this data, making all this data available. Without that, also this resource wouldn't have been possible. And also many people worked really hard on this project. So I just wanted to give them a shout out and thank them for all their hard work. Especially Graham, who really started and led this project to finish. Mike Snyder and Mark Gerstein were very the resources that they provided made this happen, and they were very supportive. And then Rashid, he was an undergrad at Stanford when he was doing this. And I think he started when he was a freshman, and now he's starting as a PhD student in my lab. So it's been really great to work with him, and also many other students listed here and collaborators, and this work wouldn't have been possible without any of them. So really grateful, and thank you so much. I'm happy to take any questions.
Thank you so much. That was very exciting and very cool. You showed early in your slides that the distribution of motif size and repeat expansion, and you noted the peak at 4 base pair motif size. Is there a reason? Is there like a biophysical reason for why 4 base pairs is preferred, or is that just unknown?
I have no idea.
Yeah, that's what I think.
Yeah. So, you know, these kind of projects where you have this so much data and so much knowledge you're gaining, it's like every single piece requires its own project, basically, why this is happening. So initially, we just basically built this resource to people. Oh, these are the things that we are seeing, and these are the observations. And then now we can actually sit down and ask all these questions that we haven't yet get a chance to do.
That's awesome. Cool.
Can I jump in with a question?
Yes,
this might be naive because I don't really, I mean, before this talk, I knew very little about repeat expansions. So just in the sense of characterizing some of these expansions, do you see, or would you expect to see maybe similar patterns of repeats in similar cancer types, or is it just completely, you know, random depending on tissue type or whatever other aspects?
So in a single cancer type, if we see, so these are recurrent repeat expansions, recurrent meaning that the same repeat expansion was recurring in multiple patients, but we did not necessarily either look at it or see it. I can't remember now that the similar cancer types having similar repeat expansion. I mean, there are also only 160 of them. Some of these things, you know, we can't statistically be convinced to say anything. So that's why I think some of those questions we just did not look.
Yeah, that makes sense. I was just curious.
I have one other thing that I'm just really curious about, and again, it's something you kind of like flagged, but didn't have much time to say a lot about it. But the negative correlation between copy number variation and repeat expansion in like TP53. Am I getting that?
Oh yeah,
right?
the simple mutations, the
right.
10Vs and the recurring repeat expansions.
Yeah. Why is there a negative correlation?
Great question. I don't know. I think we need to
seems really real.
Yeah, so we need to first make sure that it's real. So we need to do some more analysis and also some functional studies, perhaps, to figure that out. There are some functional studies in ALS, for example, about TP53 and the repeat C9 of repeat. So perhaps similar analysis needs to be done. But it is interesting that this 5 gene shows up really significant. But we also don't have ton of, again, we don't have ton of power, right? So we have 2,500 cancer genomes. I mean, the test shows that it's significant, but I think there needs to be more to be able to say why and if.
Right. Okay, last question, I promise. So single cell RNA-seq swept through academia, and everybody had to do single cell RNA sequencing. And I know that people are starting to do single cell whole genome sequencing. Are you interested in applying this to those data sets, and how do you think that might be different?
So I think that people use it for, you know, things like clonal expansion in cancer, where you look at the single cells and how the mutations are changing from cell to cell, and then to build all these evolutionary trees, cancer evolution. I mean, I wonder if you can look at how the repeats are changing, and but I'm not an expert
across those phylogenies.
So, but I'm not sure if these tools will be powerful enough to capture it from single cell data.
Gotcha.
Yeah, so it needs to be calibrated for the noise and
Mhm.
how the data is collected. So, but let's say we have a tool that can capture repeat expansions in single cell whole genome sequencing data. I think that similar studies where they do with the SNVs or even SVs or copy number variation,
Mhm.
similar studies can be done by looking at how the repeat expansions, are they changing their size?
Mhm.
As you know, metastasize, the cancer goes through different stages, or are you contracting them, or are they, disappearing, reappearing, and when it is appearing? Yeah, so those are interesting questions, and there are some interesting mathematical problems there too.
Mhm.
I wonder how it happens in, before it becomes pathogenic, and when it
Yeah.
becomes pathogenic, and where it starts, and is these repeats, giant repeats, acquired in one single cell division, or how many
Yeah.
cells you need to have? Or is it just one mistake, or is it mistakes on top of mistakes?
Right.
Yeah,
Wow. The world is
A
vast.
lot of future work.
Yeah.
yeah. So this is it. The repeats are, really, every time I talk to someone, it's not a ton of research on them because it's very difficult to do the science on them, but
Mhm.
there are a group of people doing great things, and I mean, we wouldn't be able to do this without Expansion Hunter de novo, for example. If that tool wasn't around, that we wouldn't be able to do this. And then now I'm hoping that we develop more tools where people can do more studies, where they can look at the sizes or genotype. Is it heterozygous, homozygous, can we tell those? Yeah.
All right. Very cool. Thank you so much. Thanks for working on developing the tool, and thanks for telling us all about it.
Thank you so much for the invitation. And thank you for the platform. It's been very useful.
Great. That's good to hear. All right.
We're glad to have you.